So far from this chapter, Biomolecules Part 1, we have discussed the introductory part of the biomolecules. Revise, take the reference of your class notes and keep responding. Okay. In the beginning we have conducted what? Analysis. Before that we had that comparative analysis between what? A living tissue and an earth crust. And how you have done that chemical analysis by using what chemical? Trichloroacetic acid. Okay. And tell me what will be the molecular weight of biomacromolecules? Biomacromolecules. Biomacromolecules. I asked you what will be the molecular weight for biomacromolecules. More than 800 Daltons to 1000 Daltons. In which fraction are they extracted? Acid insoluble pool, or that is retentate. And in acid soluble pool what you will get? Biomicromolecules or the cellular constituents. What will be the molecular weight? Less than 800 Daltons or 18 to 800 Daltons. Okay. What is the exception with the case of lipids? Exception with the case of lipids. Their molecular weight is? Their molecular weight is less than 800 Daltons, but then you observe them in the retentate or acid insoluble pool because they form micelles because of the cell membrane rupture. Okay. And after this what you have dealt with, ma? Primary metabolites and secondary metabolites. Give me some examples of secondary metabolites, for which I gave you a mnemonic also. If NCERTs are there in front of you, once recall. Secondary metabolites. First is pigments, include carotenoids, xanthophylls, anthocyanins, this comes under. After that alkaloids include morphine and codeine. After this terpenes include monoterpenes, diterpenes, sesquiterpenes, polyterpenes. After that essential oils include lemongrass oil. After that toxins include abrin and resin. After this lectins include concanvalin A, that is lecithin, right? After that drugs include vinblastine, vincristin and curcumin. After that polymeric substances include rubber, gums and cellulose. Still here it is clear, Nama? Any doubt anywhere? No, right? Now, so after primary and secondary metabolites, we have started with biomacromolecules, where we have clearly dealt with carbohydrates, which are also called as saccharides. Based on the number of carbon atoms, they are being divided into monosaccharides, disaccharides, polysaccharides. Monosaccharides examples include beta? Glucose, fructose and maltose. All the monosaccharides are reducing sugars. Accepted?
Yes.
Why?
They are free aldehydes and ketones.
Very good Raghav. They have free aldehyde and ketone groups. And after this we have looked into what? Examples of monosaccharides. Tell me the examples of monosaccharides, ma? Glucose, fructose, mannose, galactose and hexoses. Okay. Disaccharides examples? Sucrose, maltose, lactose, trehalose. Tell me the composition of lactose. Lactose is made up of
Galactose plus
glucose and galactose. And maltose? Glucose and glucose. And one more disaccharide? Trehalose combination? 2 glucose units. Okay. And then
maltose and trehalose are same,
no. Yes. After that we have learned through trisaccharides and tetrasaccharides, and then about polysaccharides. So complete thing is clear to you, know? Recalled and referred everything. Now in today's class we are going to exercise proteins and lipids. Proteins is a polymer of amino acids. Unlike is a heteropolymer, not a homopolymer. Sorry. because it has different functional groups, that is R group. Okay. Now, amino acids, here if you see, we have two types of amino acids. Okay, what are they? Hydrophilic polar amino acids and hydrophobic non-polar amino acids. So, hydrophilic polar amino acids, here we have positively charged, negatively charged amino acids, negatively charged and positively charged. Hydrophilic polar means there will be hydrophilic polar, which are water, which will attract water molecules like. So, here if you see, negatively charged includes what? Aspartic acid and glutamic acid. Positively charged hydrophilic proteins or amino acids include what? Lysine, arginine, histidine, serine, threonine, aspargine, glutamine and cysteine. Okay. These are the molecules which are included under what? Hydrophilic polar positively charged. Of them, lysine and arginine, just a second, of them lysine and arginine, these are the major compositions of what proteins? Histone proteins. Histone proteins. Fine, lysine and arginine are the major composition of what? Histone proteins. Next, hydrophobic non-polar, okay, includes aliphatic side chain or aromatic side chain. Aliphatic side chains includes what ma? Glycine, alanine, valine, leucine, isoleucine, proline and methionine. Aromatic side chains include phenylalanine, tryptophan and tyrosine. So, to remember these aromatic amino acids, negatively charged, positively charged is easy to you. Okay. So, fix these things in your mind and aliphatic side chains will automatically you will remember this. Fine. So, from protein part, so far what we have learnt ma, protein is a heteropolymer because each amino acid will have a different functional group. Okay. Thanks for correcting. And what is a protein chain made up of? Amino acids. So, what are the building blocks of protein? Amino acids. And here you have to remember a flow chart, that is amino acids are hydrophilic and hydrophobic. Hydrophilic positively charged and negatively charged. Hydrophobic we have aliphatic side chain and aromatic side chain. Clear with this? Okay. Next you listen. So, these are the things which are given in your NCERT, okay, wherein glycine has a hydrogen group, alanine has a methyl group in the R group and hydroxymethane group in the serine. Okay. To remember this essential and non-essential amino acids, I will give you a mnemonic. Yeah, PVT, Tim Hall. Here you see, you remember only essential amino acids. What are the essential amino acids? Those which we are taking as nutrition, okay, those which are supplemented from outside the body. Non-essential which are already present, okay. So, essential amino acids include what? Phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine and lysine, okay. PVT, Tim Hall, this you remember for what ma? Essential amino acids, take down. This is for essential amino acids. Essential amino acids, fine. Proteins are heteropolymers because each amino acid has a different functional group, and we have hydrophobic, hydrophilic, hydrophobic negatively charged, positively charged, and hydrophilic we have aliphatic side chain and aromatic side chain. And here comes the essential, non-essential amino acids. These structures mandatorily you have to fix it in your mind because they are from your NCERT. Glycine with hydrogen group, alanine with methyl group, serine with hydroxymethane group. Done ma? Next. Here you see the concept of zwitterion is applicable to amino acids. What is a zwitterion actually? Zwitterion is nothing but any molecule with a functional group, having both positive charge and negative charge. Okay, any molecule with a functional group with at least having one positive charge and one negative charge. So, the net charge of this zwitterion will be what? 0. Okay. So amino acids, when they are at the neutral pH, they will have one positive charge and negative charge, okay, in the functional group, and a functional group, right? Carboxyl functional group, amino functional group is there with positive and negative charge. At neutral pH, this is called as what? Zwitter ion. Raghav, sit straight, listen carefully, okay? So this is what, ma? Zwitter ion. When you increase the pH of this Zwitter ion, when you increase the pH from neutral pH, when you increase the pH, it turns into an anion, okay, and the net charge will be what? -1. When the pH is reduced from the neutral pH, when the pH is reduced, this turns into what? Acidic condition, and the net charge will be positive charge. Clear, right? So this is the concept of Zwitter ion. Many a times asked question. Okay, done. Next. Here you see, this is about peptide bond, how two amino acids are connected to each other. Just now we have learned that each amino acid will be having an amino group and carboxyl group. Amino group of one amino acid will be connected to the carboxyl group of the next amino acid. Okay, here you see, ma'am, the peptide bond between amino acids is formed when carboxyl group of one amino acid reacts with amino group of the next amino acid. Fine. So this bond also will eliminate what? Water moiety. Water will be eliminated. Same like that you see in the glycosidic bond. Okay, water moiety is eliminated. As such, a chain of amino acid is formed. That chain is called as what? Protein chain. Based on the formation of protein chain, we again have what? 4 different types of structures in the proteins. The first amino acid is called as what? N-terminal amino acid, and the last amino acid will be called as what? C-terminal amino acid. So protein structure is at 4 levels here: primary, secondary, tertiary and quaternary. 4 levels of protein structure is provided. So here you see the sequence of amino acids. First we will observe primary structure. Okay, primary structure. Primary structure is chain of amino acids, ma. So here in the primary structure in that protein chain, what you can analyze is positional information of amino acids. Okay, like which amino acid is present at what position, you can understand through what? Primary structure of amino acids. So here you see the sequence of amino acids, that is the positional information in a protein, which is first amino acid and which is the second amino acid, is called the primary structure of protein. Clear? These are the direct liners of your NCERT, because when you go through your NCERT reading again, you should not find any confusion. That is the reason I have picked up the direct NCERT liners. A protein is imagined as a line. The left end represents by the first amino acid and the right end represents by the last amino acid. Okay, so this will be the first amino acid and this is the last amino acid. You have to remember the ends. Left end is the first amino acid, right end is the last amino acid. At the first end, what will be free? Free amino group is present. At the last amino acid, free carboxyl group will be present. Okay, first amino acid also called as N-terminal amino acid because it has free amino group, and last amino acid will have C-terminal amino acid because it has carboxyl group. Understood the primary structure, ma? In primary structure, what you will get to know? Positional information of amino acid. At left end, you will have first amino acid, that is called as N-terminal amino acid. At last, you will be having C-terminal amino acid, that is at the right end. Okay, clear? Any doubts anywhere, ma? Next. Secondary structure, we have 2 types of secondary structures. Those include alpha helix and beta plated sheets. Secondary structures includes alpha helix and beta pleated sheets. Listen, so in proteins only right-handed helices are observed. Alpha helix, in an alpha helix what happens is, first you listen here, carbonyl group of one amino acid will be connected to the 4 down amino group, 4 down amino acid. Of which group? Amino group. Okay, So carboxyl group of one amino acid is connected to the next amino acid's amino group. Same the case here, carboxyl group connected to the amino group of 4th down amino acid. As such, it forms a ribbon like structure. What are these bonds? Hydrogen bonds. Okay, what are these bonds, ma? Hydrogen bonds. Yeah, 2 down the chain. 4 down the chain. Yeah, it is there. I am just saying like the amino acids are connected to 4 down the chain. That is the reason it is forming a helical ribbon like structure, that is alpha helix. Okay. In beta plated sheets, 2 separate amino acids will be there. The carbonyl group and amino group of 2 polypeptide chains will be connected. This is also what? Hydrogen bond. Look into the theory also once. Alpha helix in an alpha helix, the carbonyl of one amino acid is hydrogen bonded to amino group, is hydrogen bonded to the amino hydrogen of the amino acid, that is 4 down the chain. Fine. This pattern of bonding pulls the polypeptide chain into what? Helical structure that resembles to a curled ribbon. That is about alpha helix. Next is beta plated sheet. In beta plated sheet, 2 or more segments of a polypeptide chain, they line up next to each other, okay, forming what? Sheet like structure. They are together by what again? Hydrogen bonds. Clear with the secondary structure? Secondary structure includes what? Alpha helix and beta plated sheet. Here in alpha helix, hydrogen bond is in between 4 down groups of carbonyl and amino group. Here it is in between 2 polypeptide chains. Okay, that is in beta sheet. Next, here it is tertiary structure. Tertiary structure of protein is like a woolen structure. So here you see, in addition to long protein chain is also folded. It is also folded into hollow woolen ball, which is giving rise to what? Tertiary structure. This gives us 3-dimensional view of a protein. Fine. Tertiary structure is absolutely necessary for many of the biological activities of the proteins. Okay. Tertiary structure only will be active. Fine. R group interactions include what? So here in the tertiary structure, what are the bonds that are present? Unlike the hydrogen bond you have seen in the secondary structure, here you can also see hydrogen bonding, ionic bonding, dipole-dipole interactions and hydrophobic interactions. Clear, right? So tertiary structure includes what bonds, ma? Hydrogen bonding, ionic bonding, dipole-dipole interactions and hydrophobic interactions. Next is quaternary structure. Quaternary structure. So in quaternary structure, best example taken is what? Hemoglobin with 2 alpha subunits and 2 beta sheets. 2 alpha subunits and 2 beta subunits, and here you can also see alpha helix and beta plated sheets. Okay. So it has 2 alpha subunits and 2 beta subunits. So some proteins are an assemble of more than one polypeptide chain in which it can have primary, secondary and tertiary structures. Hemoglobin is the best example for what? Quaternary structure. Adult human hemoglobin will be having how many subunits? 4 subunits. Clear with primary structure, secondary structure, tertiary and quaternary? Okay, clear, right, ma? Next. Here we can see different types of proteins. Again, this is your table, okay, of NCERT. So look at here carefully, ma. Collagen is the abundant protein after rubisco, okay, in the animal world. In animal world, which is the abundant protein? Collagen. This serves as what? What is its functions? Intercellular ground substance. And all enzymes can be proteins, we have learned. Trypsin is what? An enzyme. This is also a protein. Hormones are also protein, for example insulin. Antibodies are also proteins which fights against the infectious agents. And receptor is also a protein that senses reception. through either smell, taste and hormones. Next, GLUT4 enables what? Glucose transport into the cell. Okay, this is a channel protein. Fine. As such, proteins will have diverse functions in our body. Fine. Now you see the functions of proteins.
GLUT4 is a mechanism.
No, that is a channel. See, when we are learning in cell membrane, we have learned about carrier proteins, channel proteins. Okay, these types of proteins we have learned, right? GLUT4 is what? A channel protein that transports glucose. Okay. Next, functions of proteins. Some proteins transports nutrients across the cell membrane. Okay. Some are hormones. Some proteins will transport the nutrients across the cell membrane. Some are hormones like insulin, some are enzymes like trypsin, and some will fight against the infectious agents like antibodies. Collagen is the abundant protein in the animal world. Rubisco is the abundant protein in the plant world. Rubisco is synthesized from? Rubisco is synthesized from double stranded circular DNA, which is present in the chloroplast. We have discussed, right? Okay. So, this is about proteins beta. Any doubt anywhere? From proteins, what did we learn? They are heteropolymers because they have different amino acids with R chain, R group, and then we have learnt about about different types of amino acids. Okay, hydrophobic, hydrophilic. Okay, hydrophilic polar amino acids can be positively charged or negatively charged. Tell me few negatively charged polar amino acids. Aspartic acid and glutamic acid. And then we have learnt about hydrophobic non-polar amino acids, which include aliphatic side chains or aromatic side chains. Tell me those amino acids which have aromatic side chains. Tryptophan, phenylalanine, tyrosine. Okay, tyrosine, tryptophan, phenylalanine. After this, we have looked into different structures of proteins: primary structure, secondary structure, tertiary structure and quaternary structure. Primary structure will just give you the positional information, wherein left end is N-terminal with amino group, right end is C-terminal with carbonyl group. Okay. After this, we have learned secondary structure. In primary structure, only peptide bond is included beta. In the secondary structure, along with the peptide bond between the amino acids, we also have hydrogen bonds. Okay, in alpha helix, if you see, hydrogen bond is in between carbonyl group of one amino acid and amino group of 4 down amino acid. Okay, and then we have beta plated sheets where hydrogen bond is in between two chains, two helical chains. After this, we have tertiary structure. In tertiary structure, along with the hydrogen bonding, we also have few more bonds. What are they? Ionic bond, dipole-dipole interactions, hydrophobic interactions. Okay, wherein because of these bonds, they are forming what? Hollow woolen structure. Quaternary structure of the protein, best example is what beta? Hemoglobin, adult human hemoglobin, wherein it has 4 subunits, 2 alpha and 2 beta. In the quaternary structure, you can come across the primary, secondary and tertiary structures. So, here what are the bonds included? Again, all the bonds which are present in the tertiary will be included in the quaternary as well. And we have seen the diverse functions of proteins. Protein can be an enzyme, protein can be a transporter, protein can be an antibody, it can be a collagen which is an intercellular ground substance. It can also be a receptor. Okay. So, that is the diverse composition of protein beta. So, all that with respect to protein is completed. Now, nucleic acids. So, nucleic acids, you see, nucleic acid we will call it as what? Polynucleotide chain. Polynucleotide chain. So, this polynucleotide chain, if you see, together with polysaccharides and polypeptides, these comprise the true macromolecular fraction, okay, which is observed in acid insoluble pool, okay, or in the retentate. Fine. So, here nucleotides are what? Building blocks of nucleic acids. Okay, what are the building blocks of polysaccharides? Monosaccharides. Okay, what are the building blocks of proteins? Amino acids. What are the building blocks of nucleic acids? Nucleotides. Fine. So now we have to see how a nucleotide is composed of beta. Nucleotide is composed of sugar and phosphate. See, first nucleotide is composed of sugar, yeah, right, I'll tell you. Nitrogen base first, nitrogen base plus sugar, phosphate group, okay, which is phosphoric acid. This is negative. You can take it as H3PO4. So this will give the phosphate group, which is negative. Because of this only DNA or RNA is getting what? Negative charge. So here first nitrogen base and sugar will form a nucleoside. Fine. Along with this nucleoside, to this nucleoside if phosphate group is added, then you will call it as what? Nucleotide. So here is an example, you see. Sugar will be what? Sugar will be either ribose or deoxyribose. I already told you what is ribose and deoxyribose. Both ribose and deoxyribose are pentose sugars. If at the second carbon OH group is there, then you will call it as what? OH group is there in the second carbon, then you will call it as? Which is sugar? I said if at the second carbon sugar is OH group is present, then what type of sugar it is, I told. Confirmed, huh? Fine. So these are the rough structures which I have drawn. Tell me of these two, which is ribose sugar and which is deoxyribose sugar. This left side one is what? Right side one is ribose sugar, means it has OH group. So here in this picture it is deoxyribose sugar, means at second carbon there is no O group, okay. It is deoxy. Along with this nitrogen base is added, that is adenine group, okay. When the nitrogen base adenine is what, ma? Purine. Nitrogen bases are of two types, we have purines and pyrimidines. What are they? I have to give you a rough composition. Take down parallelly. Nucleotide is formed by what? Sugar, nitrogen base and phosphate group. So here you see sugar can be what, ma? Ribose, yeah, ribose or deoxyribose. Nitrogen base can be what? Purines, yeah, purines and pyrimidines. Purines can be adenine and guanine. Pyrimidines can be cytosine, thymine and uracil. Fine. Of these three, in DNA, thymine and cytosine are present. In RNA, yeah, uracil and cytosine are present, okay. And this will form what? A nucleoside, okay. Now I will be giving you a terminology. If ribose sugar is there, okay, sugar is ribose plus nitrogen base. What nitrogen bases are present here? Adenine, guanine and in RNA and you will call it as what is adenosine, guanosine,
cytidine, uridine,
yeah, guanosine, uridine, cytidine. When phosphoric acid is added to it, phosphate group is added to it, this turns out to be what? A nucleotide. Okay? Addition of phosphate group. Adenylic acid.
guanolic acid,
Yeah, adenylic acid. Guanylic acid. Uridylic acid. Cytric acid. Cytidylic acid, sorry. Okay, cytidylic acid. So these are what? The compositions of sugar, nitrogen base and a nucleotide. Any doubt anywhere? No, right? Now you see. If sugar is deoxyribose, to this you add deoxyadenosine, deoxyguanosine, deoxythymidine, deoxycytidine. Same thing, deoxyadenylic acid, deoxyguanylic acid, deoxythymidilic acid, deoxycytidylic acid. Clear, right? Now you have to know the bonds beta. Okay, bonds, what a nucleotide consists of, what bond, that you have to know. So as you know, sugar has what? Pentose sugar, it is pentose sugar, right? So first in between C1 carbon of the sugar, what is attached? Nitrogen base is attached. If this nitrogen base is a purine, purine means adenine or guanine, purine, that is adenine or guanine, then it will be C1N9 bond, C1N9 glycosidic bond. So what is the bond that is formed between nitrogen base and the carbon of the sugar? Glycosidic bond. What is that bond, ma? This is glycosidic bond. Fine. And C1 of the sugar, C1 of the sugar, and N1 will be connected if it is pyrimidine. If it is pyrimidine, what are pyrimidines here? Uracil. I am talking about DNA. So what it will be? Thymine and cytidine, cytosine. Good. So it will be C1 and N1 bond. C1 and N1. Fine. So first carbon of the sugar is attached to the nitrogen base. If it is a purine, it will be C1N9 bond. If it is a pyrimidine, it will be C1N1 bond. Okay, that is N-glycosidic bond. Fine. Now you see, Raghav, sit properly.
and both are glycosidic bonds only.
Huh, that is glycosidic bond. Any doubt, ma? Glycosidic bond. So this forms out to be what? A nucleoside. Now the fifth carbon of the sugar is connected to what? Phosphate group. By what bond? Is ester bond. This is called as phosphoester bond. Phosphoester bond. Fine. What is this bond, ma? Phosphoester bond. So the first carbon of the sugar is connected to the nitrogen base. If it is purine, it is what?
And
C1N9 glycosidic bond. If it is pyrimidine, it is C1N1 glycosidic bond. Okay. And to this nucleoside, phosphate group is attached to fifth carbon. This is called as what? Phosphoester bond. These are the diagrams of your NCERT. You can clearly see ribose sugar. You can easily confirm looking into the number of carbons, right? C1, C2, C3, C4 and C5. And at second carbon there is OH group. Undoubtedly it is what? Ribose. If at second carbon oxygen is not there, then it will be deoxyribose. And here you see beta. Adenine, which is a purine, is binded to which sugar? Adenine is binded to which sugar? Ribose sugar. Very good. Okay. and to this ribose sugar if phosphate group is added, then it is adenylic acid. Okay, adenine at C1 carbon and phosphate group at 5th carbon. Fine. Now you see pyrimidine, uracil is the pyrimidine, right? This uracil is nitrogen base is binded to which sugar again? Ribose sugar. Will form uridylic acid. Clear, right? Any doubt anywhere? No, right, ma? Next. So these are different nitrogen bases we have: adenine, guanine, thymine, cytosine and uracil. To these nitrogen bases you should be clear with what? IUPAC names. Okay, you have to be clear with IUPAC names. Look into that clearly. Adenine is also called as 6-aminopurine. Fine. Guanine is also called as 2-amino-6-oxypurine. Cytosine is also called as 4-amino-2-oxypyrimidine. Take down parallelly. Thymine is also called as 5-methyl-2,4-dioxypyrimidine. Uracil 2,4-dioxypyrimidine. Their presence you already know in DNA, RNA. If it is done, let me know. Done? So with this I will stop today actually. Okay. In our next class we will discuss the bonds involved in a polynucleotide chain, in a double stranded nucleotide chain. We will clearly look into Watson and Crick's model of DNA, double helix model. We will also discuss Chargaff's rule, okay, and then we will also look into the RNA and types of RNA and lipids. Fine? Clear with today's class, no? Yeah, that's all, beta. Thank you. If you are having any doubts, you can ask me. Any doubts in either all the structures of proteins or here? Yes?
Uracil is similar to thymine because 2,4-deoxypyrimidine is similar. Only 5.
It has methyl group, no? But at 5th carbon, it has methyl group. Rest it is similar. Okay? Thymine is having methyl group at 5th carbon. If not, it is also what? 2,4-dioxypyrimidine like uracil. But in addition to that, it has what? 5th carbon methyl group. Good. Okay?